their questions someday. There's a clicker question coming soon, so you might want to grab your toys. All right, so this is coevolution. This is part of the stuff that I really think is fun. I realize that what I think is fun may not always translate. We're going to finish up with a special version or a special topic in mimicry, in coevolution called mimicry. I'm going to turn my phone off. And then we're going to kind of move up in scale. So we've been talking about species-species interactions, and then we're going to ask how does that translate into making diversity, building diversity. So we're going to kind of move up the level of organization. All right, so mimicry is a really fascinating interaction between species that's captivated people since before we really had a very good concept of evolution. And it's this observation that things tend to look alike, species look alike. I'm going to give you one really narrow example of mimicry that's maybe the most commonly thought about, but realize that this is, I'm kind of carving out this whole world of cool stuff like aggressive mimicry and sexual mimicry and males who pretend to be females for various reasons. I'm going to talk about predator-prey mimicry, or what's often called protective mimicry. The basic idea is it's three players. It's called a tripartite system. There's a predator looking for a meal, and there are two things it might eat. One of them is nasty, that's the model, and one of them tastes good, that's the mimic. So a model has some way to deter a predator. It tastes bad, it has poison, it has spines. And then the mimic does something that confuses the signal that the model gives to the predator and uses that same signal and gives it to the predator and gets protection from it. So there's a lot of dynamic in that simple little two-way interaction. The most famous one probably is monarch butterflies. Here's a monarch. Monarchs get their defenses when they're babies. They are caterpillars. They grow up on milkweed. They sequester cardiac glycosides. Cardiac glycosides can kill predators. They make them puke. So there's a famous picture of a blue jay yakking up a monarch butterfly caterpillar that it just ate. Depending on the host plant, they get more or less toxic. There are a bunch of butterflies that look similar. This is a viceroy. You can obviously see the difference, right? So sometimes mimicry is really, really close. The difference has to do with this absence of a white spot here and the presence of a white spot, and then this cross vein on the hind wing that monarchs don't have. Right? That was obvious to everybody. So imagine you're a predator, you're trying to get a meal. Right? What happens in a mimicry system is a couple of things. The predator learns a signal, so the individual learns this is bad. So every time it eats a monarch, it says, ooh, orange and black butterfly, man, I'm not doing that again. And every time it eats a viceroy, it thinks, that wasn't so bad, I can eat that. So there's a learning and forgetting process. So the predator learns, and what's evolving is the color pattern of these two things signaling to the predator. So in most systems, and there are some exceptions, but in most systems, the predator learns the avoidance. The mimic is then driven to look more like the model because it's protected when it looks like the model. Make sense? So if you think about the selection, what's happening is selection is kind of directional on the mimic to become more model-like, but the model is also getting pushed away, and I'll come back to that, and the predator learns. All right, so here's the question, first question of the day. All right. Wasps, you know wasps are yellow and black banded, right? So you don't grab things that sting you. But imagine you're a predator and you're trying to get a meal this morning. Which of the following two things are you going to grab? So everybody look up. Which one do you grab? It flew away. All right. So A or B? Are you going hungry? Are you going to grab both of them? Did you pick A or did you pick B? I'm going to let you be a lucky predator and look at them closer. So, Who do you pick? A, B, both, or neither? This is what a predator has to do to get by in a day in the world.
되게 엔트나만 좀 다른 것 같아요.
So this is a really interesting psychology experiment. Okay, so I just learned a lot about it. 50% of you aren't willing to take risks. So I tell my lab, this is the lab motto in my research lab, err boldly. Don't be afraid to make mistakes. Just go out there, try it. If you screw up, who cares? In this case, there's a cost, so maybe you want to be a little more cautious. Only 7% of you are really gutsy and would grab both of them. I'm sorry about that. This turns out to be a fly, right? No stinger. This is a wasp, a yellow jacket. The difference, if you know your insect taxonomy, or if you take entomology at Mountain Lake in the summer, flies have one pair of wings, wasps have two, and they have a slightly different kind of antenna. But admittedly, if you're a bird looking for a meal, you're probably not going to go to that detail. Right? The point is, mimicry can be intensely close. Right? You can get what are often called almost perfect mimics. Okay. Oops. Too many programs at once. All right, so let's go back to this learning thing. You just learned that, you guys have no guts, that you're timid and you want to live a safe life. All right, so this is another famous mimicry system from the Smokies. This is a red-cheeked salamander. This is a thing called the imitator salamander, very creative name. This one has a nasty skin secretion. You can kind of see this white goo, and it burns mucous membranes. If you put one in your mouth kind of goes, scrunches up like a dried apple. This one is like the Big Mac of the forest, right? Everybody eats it. But birds learn that the red cheek signals this nasty secretion. So again, predator learns, every time it eats the red cheek, it learns red cheek bad, red cheek bad. Every time it eats an imitator, it thinks red cheek good, okay? So there's a number game. We talk about it like frequency dependence, it's really numbers. The more often that it encounters the negative reinforcement, the nasty salamander, the better that learning is, the higher the avoidance of that pattern. And then every time it encounters an imitator, it says, okay, that's okay, I'm going to eat that thing. So there's a dynamic of learn, forget, learn, forget. And so the prediction that comes out of it is we should always see more models than we should see mimics. Because it's a learning, forgetting process. If the predator is being taught, it has to be taught more often to avoid than taught to attack for this system to work. So if you count up the numbers of salamanders in the forest or butterflies of different types, you almost always see the models are more common than mimics. Otherwise, the advantage to being a mimic disappears. And so that frequency dependence holds the numbers in balance. Okay? Okay, so these are examples of a thing we call Batesian. This is, for whatever reason, the hardest name for classes to pronounce. This is not Batesian. It's named after a guy named Henry Bates, so it's Batesian. Or Mullerian, named after this guy named Fritz Muller, both operated in the 1860s. They're kinds of ends of a spectrum. So the things I described are Batesian, meaning the definition of Batesian mimicry is a model that's nasty and a mimic that's okay, good to eat. Mullerian is different because both species are models and mimics. They both reinforce the negative numbers game. So right away you should see that there are differences in the dynamics. That in the Batesian mimicry system, you've got one teacher and one, I don't know which that makes you forget. In a Mullerian system, both species teach. Right? So the selection dynamic's a lot different. Everybody get the difference? It's not absolute. That's the other thing to keep in mind. One end of the spectrum, you've got Batesian. At the other, you've got Mullerian. And then most of the time, there's like, some things taste bad, but if you were really hungry, you'd eat them. Right? So there's this thing called a palatability spectrum. So what really separates those ends of the spectrum is selection, the kind of selection that operates. In Batesian mimicry, the mimic is being selected to converge on a model. Right? There's a signal, predator learns the signal, selection pushes the Batesian mimic to look more like that model. So that's directional selection on the mimic. And as the mimic gets closer, what happens to the predator? It starts to learn that that's not such a bad thing. So there's selection on the model to get a little bit different. And we call that chase away. Right? So mimic gets closer to the model in space, in what it looks like, and that generates selection on the model to move further away in space. So the number of red spots or number of black bands. Right? Contrast that to Mullerian mimicry, where both prey types teach. Now, predator learns when it attacks the model, predator learns when it attacks the mimic. What's the best color pattern to be? The most common one. Right? So if you're a prey, you're trying to deter a predator, is it good to be the one that teaches the predator? Do you want to be the butterfly that teaches the predator? What happens to the butterfly that teaches the predator? It's dead. So this works because other individuals have died while you get away, you kind of slide by. Right? So selection is going to favor the color patterns that look most similar in both species. So that's the big difference between these two kinds of mimicry. In Mullerian, everybody teaches, so selection pushes all the color patterns to be really common. So if you look back at the pictures from last time of those heliconius butterflies, that's what happens in those big complexes of South American butterflies. They all move towards one common morph. Does that make sense? It's bad to be the prey item that teaches the predator. You want to be the prey item that is encountered by the predator that already learned. Okay. Here's an example of Mullerian mimicry, another local one. These are millipedes. You may or may not. So what I've learned is that students have no concept of what lives in the forest. Most of you have probably never thought about a millipede. There are these little detritivores that eat rotten organic matter. All of them make cyano compounds, so things that are related to cyanide. They smell really nasty. I guarantee you would not put one in your mouth. Birds also don't want to put them in their mouth, and they can actually kill the birds. Right? So here are localities around Southwest Virginia, Kentucky, Tennessee, and where you find one pattern, where you find differences in patterns. So here, this one's mostly yellow, down here it's got red legs. The other species that lives in the same place matches that color pattern. Right? So you can go across the southeast and you find this matching. All of them are toxic. This is Mullerian mimicry. So this is work that was done by a guy named Paul Merrick, who's now at Tech as a faculty. Okay, so here's another question. Think about just Batesian mimicry now. It involves 3 players. We're talking about coevolution. Which species are actually co-evolving? Predator and mimic, predator model and mimic, all 3 of them, or just the prey types? So this is a little deeper than do you get stung or not, but see if you figure this out. Now you got quiet. Yeah. So that's a good question. Learning a heritable trait. Learning could be, but the actual avoidance is not. The avoidance is learned by the individual. So every individual has to learn separately. Be much more cautious about answering. Okay, you're pretty good on this. So most of you are on the path, right? And that makes me feel good because it means you're getting it. So the answer is D, the model and the mimic. Why isn't it any of the others? Because the predators, the individual predators, have to pick up that skill. So there are some examples of systems that involve really deadly prey where it looks like the predators evolved in response to the mimic signal, coral snake mimicry. But in most cases, what I want you to pull out is that the predator's not actually evolving in most cases with the prey. That's an individual learning thing. But the model and mimic, that's that chase away selection, that's where the coevolution is going on. You guys are pretty on top of that. Okay, all right, so we're gonna move up the scale. All right, so these are, you'll see this term written down in a couple slides. This is, these are then examples of what we often call specific coevolution, where two species interact and there's reciprocal selection between two or maybe more, but really tight coevolution. We're gonna move up to what happens in terms of groups and diversity when coevolution goes on. And so if you think about that coevolution as building sort of species relationships, now if we can diversify one of those species, what should happen to the other? Maybe it should go along with it. All right, so co-speciation is a slightly different term than co-evolution. So speciation means splitting the tree, bifurcating events, or coevolution doesn't necessarily generate diversity. So this is a cartoon version of two trees. Pocket gophers on your left, lice on your right. Lice are parasites of pocket gophers. And what you see when you compare, often when you compare hosts and parasites, is a pattern like this, where the species topology of the tree, the branching pattern, matches up pretty closely to what the host has. So if you track these branches, right? Here's a branch, here's a branch. There's an older branch, matches up an older branch. So co-speciation predicts that you get this matching pattern of topologies. And for a long time, people took that as evidence of evolution, coevolution, right? How else would they co-speciate if they weren't co-evolving? Right? But there are actually a lot of reasons why we might see that pattern that have nothing to do with whether the parasite and the host are really exerting reciprocal selection on each other. So the obvious, simplest one that's not very interesting is maybe hosts and parasites are evolving in response to a third variable that's abiotic, and they just both happen to be doing it the same way. So let's say that the lice on the coat and the pocket gophers are both responding to mean July rainfall, right? So the amount of water in the summer. If they evolved in response to that third variable in the same way, you get this pattern where they match up. And that doesn't mean that they have co-evolved, that there was reciprocal selection. They just happen to match the same pattern. The second one is a little more interesting, and that is that maybe there's another speciation force out there, vicariance is the easiest one to think about, that causes the two groups to evolve in the same way or to speciate the same pattern. So maybe they live in a contiguous part of the eastern US, and a new river forms and it splits the distribution, and the lice on the southern side of that river are isolated from the lice on the northern side, and so are the gophers. So they become geographically isolated again for some reason that doesn't have to do with their parasite-host relationship. Right? The third is really what we're trying to get at, and that's host-induced vicariance. So lice live on gophers. Gophers are like the islands for the parasite. And if parasites can only move back and forth from one individual to another when they come in contact, what happens if we split the gophers? We end up splitting the lice. Maybe the gophers are speciating in response to, I don't know, seed type that they're feeding on. As those lineages split and there are fewer mating events between two groups because of reproductive isolation, the parasites can no longer mate, right? That becomes host-induced vicariance. So not vicariance because of some other outside factor, but because of that relationship. Right? And as they become more different, the parasites also become more different. And that's what we're trying to get at with this concept of co-speciation. Right? So, punchline, the patterns matching are required for coevolution to generate that matching pattern of diversity, but they're not sufficient. They don't imply by themselves that coevolution happens. Okay. An example. I'll give you a couple examples. So these are lice that live on bird feathers. I hesitate to say that lice are really cool, but these lice are really cool. They're long-bodied. They live, if you've ever looked up close at a bird feather, it's got a central vein, right? And then it has barbs that come off on an angle. And the barbs have they touch each other. And these feather lice come and sit in between those barbs. And so they have to match the size of the barbs of whatever species they live on. When you've seen birds all do this with their beaks and they preen their feathers. Preening is cleaning off the lice and the lice eggs. So what they're doing is taking their bill and scraping their feather. So which lice survive? The lice that can hide in between the barbs. So there's really, really strong selection for phenotypic matching on the barb size and the louse size. So this is a cartoon version, I'll show you the data in a minute, of work that a guy named Dale Clayton has done at University of Utah. Here's a phylogeny of pigeons on the left. Here's a phylogeny of parasites. What you often find when you look at these is some pattern of matching like we would predict with co-speciation, except where it's not. And that's a host shift. So here's parasite species B actually is able to infect bird species D. So B is shifted onto D. Right? So we get a pattern of co-speciation, and then every once in a while some species manages to get onto an unrelated host and colonize it and spread. Right? So that's a host shift. What that looks like in a real phylogeny is this mess down here. And you can see that in parts of the phylogeny, you've got this nice pattern of co-speciation pretty closely. And then in other parts of the phylogeny, you've got, species 4 up here and colonized this thing down here, and then this thing colonized this thing that's not related to its closest relative's host. So there's a bunch of mismatch. And what it suggests is that ectoparasites can jump, move from species to species, and that they are speciating in their own way, in a way that doesn't match the host. So it's like colonizing new islands, but the islands are alive. And this is, here's a pigeon. I wanted to show you this picture too, because it's not just a matter of matching the size for the bill to not be able to scrape it off. There are different colors of feathers, and the birds look and see the lice on the feathers. So there's actually camouflage too. There are dark lice that live on dark feathers and light lice that live on light feathers. There's this whole world of stuff going on birds. Birds are filthy. Don't touch birds. That's host and parasite. This is a mutualistic version of a similar thing. So now there's benefit to both species. I don't have time to really do justice to this biology, so it's a really fascinating world. Figs and fig wasps. So how many of you have eaten figs? They're delicious, right? They are full of protein. They're really good. So North American figs don't have this pollinator, so you don't have to worry too much. The basic story of figs and fig wasps is here's your fig. All that juicy red stuff in the middle, those are flowers and fruits. So all the flowers are internal to that casing that you think of as the fruit. Figs, this is a generalized story. Figs have specialized wasp pollinators that live inside here. Females come in through this thing called an ostiole. They get in, they pollinate the flowers so the fruit grows. They literally carry pollen and pollinate, and then lay eggs on some of the other flowers so that some of those fruits become food for the baby wasp, and some of them are pollinated and become seeds. Right? So males emerge, they run around, they mate with females before they've gotten out of their fruit. Then they go out and they chew a hole out to the very edge of this fruit, and when the females emerge, they come out that tube, finish the hole, fly off, and go to a fruit on the same tree. So it's this really intimate, close, tight relationship. And there's really close speciation between the pollinator mutualist and the fig, because they tend not to move from tree to tree. Right? So there's all this specialization that I don't have time to really go into. That's one group of wasps. Now there's another group of wasps that cheats the system. And they have this long ovipositor. So the stingers on wasps, so the things that you don't want to grab, those are modified ovipositors, things that lay eggs. So these parasitoid wasps, or parasitic wasps, they have these long ovipositors that go through the wall of the fruit, and they just jam an egg into one of the fruits of the fig, and there you go. So they haven't done any pollinating. There's no mutualistic relationship between the fig and the wasp. They're just looking for a place to stick their egg. Okay? So you've got pollinators and you've got cheaters, or parasite wasps. So if you look at the patterns of speciation, which one would you think would be closer? Just as a guess. You have to tell me the thing for yourself. Are these guys going to speciate more closely with the figs, or are these mutualists that are more intimate? So it should be pretty obvious. The pollinators over here on the left, this phylogeny looks pretty similar between the wasps and the figs on the left. So figs, pollinating wasps. The other phylogeny are these cheaters, the parasites. Here's another group of figs. Here's a group of parasitic pollinators. That tree doesn't match up very well at all. Some of the big branches do, but the smaller branches don't match. And what it suggests is that obligate mutualism, that intimacy of interaction, creates patterns of coevolution that when one speciates, the other follows. One speciates, the other follows. The parasites are much more able to shift here. You see these cross lines and things like this where two of them infect one or parasitize one fruit. Okay, here's a question. Yeah. So, take that knowledge and think about where would you expect to see co-speciation, or what pattern would you expect to emerge that looks like co-speciation? Which of these relationships is more likely to generate? You see I loaded up all my clicker questions because I was way behind and they've all piled up at the end. We'll have to plot the response time to this. There are always like 30 people who respond before they could have possibly read the question. And then there's like this lull, and then all of a sudden everybody's got it. All right, you guys have two answers that you like. They are B and C. All right. Anybody want to defend either one of them? Put you on the spot. I like D. Right. So look at the question. Oh, sorry, B. You're awake. You're more awake than I am. Which of the relationships do you predict would look like co-speciation? Generalist predators and their prey? Yeah, no, that's the eggplant of the question. It doesn't really make sense. Mimetic butterflies and the birds that eat them? Well, we already figured out that the predators in mimetic systems aren't really co-evolving with the prey in most cases, so that's probably out. So you're left with B and C. C, these can be very tight or very diffuse interactions. The D really describes a very tight interaction. So what is vertically transmitted? It means inherited. So mitochondria are vertically transmitted from mom to offspring. Aphids have bacteria that live in their bellies that, well, not in their bellies, special organs, that are transmitted from mom to kid, mom to kid. So that's the situation that's most intimate, most close. The obligate relationship is the tightest, and would be the place you'd expect to see the pattern. Yeah.
Like we talked about how shit videos, why can't secondary split that was posting price tags? Should they always, whenever there's like should they always go the same way? Because that's
To branch the same way? Yeah.
so why can't they do specific migraines and like things like that?
So this is a really good question. So I'm trying to rephrase it. So why am I drawing a distinction between vicariance and host-induced vicariance? Because anytime you're splitting that parasite population, that's vicariance, right? Is that what you're getting at? So one of those vicariance, so let me back up. If you contrast vicariance that's, say, caused by a river, right, that splits this distribution, that's vicariance that comes from an external source. It doesn't have to do with the species interaction. But host-induced vicariance, the reason the vicariance occurs is because they're stuck on that host. Right? The host itself is the geographic distribution. So is that, yeah. So that's the distinction. Whether it's the host splitting or some external factor that splits them. Okay? All right. So again, on this theme of diversification. We talked about adaptive radiations with cichlids and lizards and how one branch explodes in time and becomes many, many different branches. Right? That's the pattern of adaptive radiation. So coevolution is thought to give a particular sort of adaptive radiation. And I'll explain that in a second. But first I want to come back to this general definition of specific versus diffuse coevolution. So again, we've been talking really about specific coevolution. When we say species A and species B, or this host that's attacked by this parasite, right? That's two species interacting with each other, reciprocal selection, gene for gene coevolution. So if you remember with the very first slide, we had the term genetic complementarity, meaning there has to be a gene in the host and a gene in the parasite, and the interaction between the two is causing changes in those two genes that are complementary. That's specific. The alternative, or other perspective, is that we can have something called diffuse coevolution. And a lot of people don't really like that term because it doesn't have all those definitional components that we talked about the first time. Diffuse coevolution happens between guilds. So in the old world, tradesmen were guilds, right? The plumbers, the electricians, this groups that did a thing. And in ecology, we have guilds, we have seed predators, we have pollinators. You can have buzz pollinators, like bumblebees and their ilk, that get onto a flower and buzz and the pollen falls out. Or you can have nectar feeders that stick their tongues way down and dig around, and that's how the pollen is dislodged. So diffuse coevolution is the idea that those guilds are co-evolving. And as, say, a flower type changes, it drags along the co-evolving guild of pollinators. So I think it's easiest to think about with something like hummingbird pollinators, pollinated plants that tend to be those long tubes, remember, that are red. So as that diversifies, it opens up new niches for that group of hummingbirds that feeds on that hummingbird pollinated guild. Right? So it's a group being attacked by another group, or mutualized. Is that a word? Having a mutualistic interaction with this other group. It can work antagonistically if it's something like a defensive compound. So a plant is eaten by a butterfly or a caterpillar, and as that group of plants evolves new defensive compounds, the caterpillars can't attack anymore. Until the caterpillars figure out the evolutionary trick to detoxify that new compound. And that's the idea that we're going to try and build off of, right? So in diffuse coevolution, reciprocal selection is broad. It's not species to species. It's guild to guild. And we don't expect these really close species-species matching of traits, right? We're not going to get that lockstep arms race necessarily. Maybe in some pairs, but not in the whole group. All right, so in diffuse coevolution, you can get this pattern of radiation that's known as escape-radiate coevolution. This is an old idea that goes back to Paul Ehrlich and Peter Raven in the 60s, and they were the first to describe this idea to explain why there's so many butterflies and angiosperms. So, and they weren't really thinking about butterflies so much as the caterpillars. And they made the observation that as plant lineages expand, they tend to evolve these new secondary compounds, poisons, that deter herbivores. And when that happens, you get a new group of butterflies that seems to speciate along with them. And so their prediction looked something like this in a cartoon version. That if you take plants on the left and insects on the right, every time you get a new innovation, so a brand new defense, that allows these things to escape their old predators. So here in orange, this is the old branch. Here are the predators or the herbivores. There's a new innovation here at the star before the green, and that allowed these guys to radiate out in a pattern that isn't matched by the insects. But then, one of the insect lineages figures out a way, evolves a way to detoxify this compound, and it radiates. So it doesn't necessarily match the speciation pattern, it's just that there's a radiation event followed by another radiation event. And then there's a new innovation up here at the blue branch in plants. That's a new toxic compound. And that allows another branch of those herbivores to radiate out. So the pattern should be, if you looked at these trees, stasis, an explosion in one, and then stasis, and slightly lagging in time probably, an explosion in the other end. So it's escape, new innovation, escape, radiate, and then the herbivore tracks it and radiates in response. And then the plant has a new innovation and escapes and radiates, and the herbivore tracks it. Does that make sense? So it's the same idea as adaptive radiation, but now we have an explanation for why, and that why has to do with the interaction between atoms. Okay, here's a busy but pretty illustrative slide of this in action. So there's really two pictures here. So right here, at the top we have a tree of we have a tree of plants. We have a phylogeny of flowers, mustards, Brassicaceae. Up here, Brassicaceae have a bunch of different toxic compounds. The names don't matter, but they're designated here in different colors. and with little symbols. So here's the origin of an indolic glucosinolate. Here's another kind of glucosinolate. Here's another compound up here between the green and the orange. On the horizontal axis, we have time, 100 million years to today. Right? And down here, we've got this group of butterflies called purines. And the prediction, if escape, radiate evolution explains some of the diversity, is that when we get an innovation, we should see a speciation or diversification rate increase in the plants. And then it should be followed by diversification of these butterflies. Right? So these dotted lines say, okay, here's the origin of this new compound. Did we see radiation in the butterflies afterwards? Yeah, looks like we did. But I'm asking you to do a squint test, which is not very good statistics, right? Like, oh, the patterns look close. I know. If you plot the diversification rates, you can see it a lot better. So on the top, you've got diversification of the plants. Here's diversification of the butterflies. Here's time on the X. We've had a long period of stasis in purple, and then boom, we get an explosion of species in green, right? That's the signal of adaptive radiation. Here's green, right? When that happens, what happens to the speciation rate of butterflies? Well, it was pretty stable for a long time, and then lining up in time with that explosion, we get a much increased rate. What happens after that explosion? Well, the diversification drops off. Right? Butterflies don't speciate for a long time. Plants don't speciate much for a long time. Then there's this new explosion here in yellow. We get a lot more species of plants. Oh, look at that. We got a lot more diversification or faster diversification of butterflies. And as that goes to a constant rate, the speciation rate of butterflies drops off. So that's escape and radiate, followed by the enemy. Escape and radiate, followed by the enemy. Okay? So there's mixed evidence that this happens. You see it at a very large phylogenetic scale, but you don't see it when you look at like a genus level, really small levels. Okay, wow, a lot of questions. Let's skip this question because it's giving me a lot of questions. All right. So we're going to do one more concept of diversifying coevolution before we move forward. So we had co-speciation, we've had escape and radiate. Now I'm going to take you back to geographic mosaics and talk about this thing that's sort of generally called diversifying coevolution. The basic idea here is that in space, we have different adaptive peaks. Those adaptive peaks cause species A to become different, right? There's selection to move up one peak or up the other peak. The geographic mosaic comes in because some places coevolution's intense and other places it's not, and coevolution favors one peak over here and a different peak over here. If that happens, then we just go back to speciation. We say, all right, if these things are selected to be different, what happens when they cross? You get an intermediate. It's not going to be very fit for either one of those places. So there's selection against hybrids, so we should get isolation. So it's just the speciation process. But it starts because those peaks are enforced by coevolution. Make sense? So here's a really nice example. This is work by Craig Bankman, who's at Wyoming now. And he's worked on these things called crossbills for a long time. Crossbills are a finch that has a weird bill that it's like a twisted tweezer, right? So when they close their mouths, their bill tips look like this. You kind of see that in the picture. And what they do is feed on conifer cones, pine cones. And they get down in between the scales on the pine cone. So here's a cone. You see how it's a little bit open? They stick their bill, they open their bill, they stick it down in that crevice, and when they close, it acts like kind of a pry bar that pries the seeds away. Make sense? So the size of the bill matches the cone, and the most fit bills are the intermediate values that match whatever the properties of the shape of this cone are. Right? And So this is, on this side, it's survival of the finch, of the crossbill. Over here, it's the feeding efficiency. So they match up. The most efficient feeders are the ones most likely to survive. It also turns out that it's good for conifers, for pines, to have thick scales because they're protected from the environment. So all else equal, pine trees would produce thick-scaled cones. Enter into the world a nasty mammal. Squirrels have been introduced to a lot of the range of these crossbills, and they eat in a very different way. They grab all the cones, they go make a midden, a stash for the winter, and they feed on these things all winter. And so they collect all the cones, and they pretty much obliterate all the seeds on the cone. The way that the trees have co-evolved with the squirrels is to produce a really heavy core that the squirrel doesn't like to carry, and relatively few seeds that are unprotected, because the squirrels decimate them. So the idea is that more of those thin scales, squirrels make mistakes, they drop some. Right? So this is screwing up the coevolution between the crossbill and the cone. Right? So in places you have squirrels, you get a cone that looks like this on the left, where you have no squirrels, it looks like this on the right. And if you're looking at your pictures going, wait, where's that? I popped this in this morning, so I'll load that up. The finches have evolved, the crossbills have evolved to match these things. So here's one with a less curved bill that fits on these things that squirrels eat, more curved where there are, vice versa. Where there are no squirrels, more cone, more dispersed scales, the bills have to be longer to pry them open. Okay? That's the biology. What happens in space? There are different conifers all over. There are squirrels some places and not others. If you map out the shape of the crossbill bill, so this is the depth and the groove width, you can see peaks all over. Each one of these cones is a thickness peak for a crossbill. So some places, they're better off if they're short and grooved, other places long and not so grooved. The hybrids are less fit. What's happening with the crossbills is their calls are becoming different. So they recognize their own local type as a mating type distinct from the place with the other kind of cone. And Craig's latest data suggests that actually the shape of the bill may change the call. So this thing that's evolving to the cone may actually affect the morphology. But it starts with coevolution, and that's why it's diversifying coevolution.